Good morning and welcome to the GRCF seminar series for the academic year 24 through 25. Our first seminar this year is being presented by Edward Sawicki. He is the Regional Sequencing Specialist with Oxford Nanopore. The title of our presentation this morning is What You're Missing Matters, an update from Oxford Nanopore and insights into whole genome sequencing. And with that, I'm going to turn it over to Edward.
Good morning, everyone. As Barb mentioned, I'm the Regional Sequencing Specialist for Oxford Nanopore. A little bit about my background. Prior to this, I was at Thermo Fisher for about a year and a half in their translational genomics unit. Prior to that, I opened a couple of labs myself. I was at Illumina before then for about 12 years. And then I also worked for the state of Maryland, and I also worked at Johns Hopkins as a med tech when I first got out of school. So I've been in genomics for about almost 20 years now. So I think next year, my genomics age will become of drinking age. So I'll be able to actually, celebrate. So if anyone has any questions during the presentation, please feel free to stop me. I like to keep things open. Let's see. Okay.
Edward,
Yes.
Folks could post who are attending virtually. They can post on the Q&A.
Okay.
They don't have the ability to speak.
Oh, okay. Perfect.
Okay.
All right. And the slides aren't moving. They're not moving. They were moving earlier when I tested it.
Bear with us for a moment. We're having some technical difficulties.
Did you try that?
Oh, there it goes.
Yeah.
So first, before I get into our updates, and I'm going to go over the updates that were recently announced at NCM, but I'm also going to first go over the technology itself for anyone who doesn't quite understand how the core technology works. So hence the name Oxford Nanopore. Our core chemistry is based off of a nanopore. And what occurs is when you perform your library prep, you're applying your adapters, you're applying your motor enzyme. And what that motor enzyme does is it allows us to slow down the rate at which the DNA and or RNA comes through the nanopore. So if you look here, here's an actual blow up of a single nanopore. And what happens is there's a tether here that will then pull the DNA or RNA down to the nanopore, and it will start to sequence. When it sequences at about 400 bases per second. So like I mentioned, I was at Illumina. I started there when they had first purchased Alexa. So it took me a little while. I've been at Oxford for about almost 3 years now. It took me a while to kind of wrap my head around the fact that we're sequencing 400 bases per second, and it's not taking a minute or up to 3 to 5 minutes to sequence one base. So the rate at which we are able to do this allows us to sequence at a rapid speed. It allows us to sequence long strands of DNA or RNA, and it allows us to basically dive into areas of the genome that other technologies cannot get into. So the technology itself is direct sequencing. You can perform PCR if you need to, but it is PCR free. We sequence native DNA. We pick up base modifications. So we detect base modifications. It's the analysis of those modifications that become difficult. So we do have a list of modified bases that we can detect. And if you have your own that you would like to be able to detect, we have a list with an R&D that we continue to work on, but you have to train the base caller to be able to determine what the squiggle means when it comes through. And when I say squiggle, as the base is passed through, there's an ionic solution. That ionic solution gets agitated and it creates the scientific term, which is known as a squiggle. That squiggle is then interpreted by our software into a base call. And this all happens in real time. So we perform real-time analysis. So say, for instance, if you take this out into the field, if there's an outbreak or if there's some type of virus that you need a fast answer for, you're able to actually perform base calling in real time and perform detection. Not only can you perform real-time analysis, but we're also read length agnostic. So we can read all the way down to 20 base pairs. I know a lot of people think that we're just a long read company, but that's not true. But we can read all the way up. I think the longest with our ultra read protocol in the hands of a customer has been about 2.3 megabases. So that's sequencing one strand that's 2.3 megabases long, just in one swath. And we're also scalable. So we have everything down from our MinION all the way up to our PromethION flow cells. We have our PromethION 48, which can do up to 48 genomes within 3 days, which I will go over. We offer a host of devices and platforms to perform your analysis needs. So we start with our Flongle. This is our smaller flow cell. This is good for like QC, small pathogens, small viruses, things of that nature. Our MinION, which is our oldest product, that is actually going to get an update, and I'm going to go over that shortly. So our MinION is great for, pathogens, viruses, smaller genomes. Those two flow cell types can run on either our Mark 1B, our Mark 1C, or our GridION, which I'll go over in just a second. These two will be being phased out. So our MinION Mark 1B was now going to come to the Mark 1D. I'm going to go over that in more detail later on. It's just an update to the device that actually holds the flow cell, and it can control temperature better. It's just a newer device. This device itself is over 10 years old. The Mark 1C is going to be replaced by the iPad. So the Mark 1D can now fit into a case that fits with your iPad. We have a partnership with Apple where your iPad now becomes your sequencing computer. So you can take that out into the field and you can do up to 3 hours of sequencing without having it plugged to any power source. The GridION's not going anywhere. That's going to be here for quite some time. We actually have Q-line and other avenues that the GridION is going to be available in, which I will go over shortly. For our larger flow cells, this is on one PromethION flow cell, you can run a whole human genome at about 30x coverage, or you can multiplex several smaller genomes. The PromethION flow cells can be run on our P2 Solo. So our P2 Solo does not have compute. You can actually purchase, we'll give you the specs to purchase your own computer, or it plugs directly into the GridION. So that way you have the ability to run both MinION flow cells, and if you want to do some single cell work, you want to do some whole human genome work, you want to do larger scale runs, you have the ability to run 2 PromethION flow cells. If we take a step further, we have our PromethION 2 integrated. That contains the compute, and it's a powerful compute, so you can do adaptive sampling, you can do all live base calling on the actual device itself. And then as we move further down the line, we have our PromethION 24, which is where you can sequence up to 24 genomes at one time at 30x coverage. And then our P48, which is just 48 samples that you can run at one time. Oxford Nanopore's goal is to provide a multi-omics approach, to be able to do both DNA and RNA sequencing. Now, I know some people have probably read about proteomics and how we're getting into proteins and how it does work on our platform, but that's still very early now, but we will eventually be getting into that arena. But if you look, I mean, you could do chromatin conformation, structural variants, SNVs, epigenetics, phasing, assembly, via DNA, via RNA. We have single cell workflow that we partnered with 10x. We could do gene expression, epitranscriptomics, splice variation, fusion transcription, and we are the only company that offers native RNA sequencing. So you're actually able to sequence the native strand of RNA and get any modified bases or anything that you currently might be looking for. As for the tech update, anyone that did listen to NCM, this is kind of going to be a refresher. So these are the slides that we went through in our Nanopore community meeting, which usually happens every December, but we had it a little early this year in September. So we have 2 instances where we have tech updates during our London Calling meeting, which is in May, and then during our Nanopore community meeting, which tends to be in December. We're enabling, as I mentioned before, a multi-omics insight from just one single platform. So with our platform, you're able to do canonical bases, native modifications. You're able to actually perform any type of look at synthetic modifications, a full host of RNA workflows that are available, and also chromatin conformation via like POR-C, which is a workflow that we offer. And with that, we're optimizing the analysis pipeline with the ultra-rich data that we provide. So for signal processing, we have base calling, and you could pick up modified bases, as I mentioned earlier. Our core tools, you could do adapter trimming, barcode demultiplexing, alignment, read correction and polishing. And for variant detection, we offer SNV detection, structural variant detection, methylation, phasing, splice variants. And when we look at structural variants, the ability that we have to do swaths of 20 to 30 kb at a time in a single read allows you to truly look at those structural variants. You're not piecing together a short read and trying to actually figure out where things go. You actually get the entire length of information. And for our bioinformatics pipeline, we have done a lot of work in the past 2 years in regards to our epitome suite. That's where we offer a lot of our workflows. So you have workflows for human genetics, infectious disease, assembly, metagenomics, target sequencing, plasmids, all kinds of other modules that you can run within our Epitome suite. So if we look here, I know years ago, I remember when I first started looking at Oxford Nanopore, this was back when the MinION was first released, the quality scores were fairly low. I think it was like 84%, 80%. We've come a long way. We've come a long way since I started. So when I started, Kit 14 was just in basically beta testing. It hadn't been fully released yet. And that was just getting us to our Q20 chemistry. So within those 3 years that I've been here, we've been able to actually bring our quality scores all the way up to Q26. And we have to remember this is a single molecule accuracy. This is the confidence that we have in that one, from the image that I showed earlier, that little video, the confidence that we have in that single base coming through and the confidence in the call that we're making for that base. Now, if we talk about consensus accuracy, we're talking about Q40, Q47, and sometimes Q60 for some of our bacterial genomes. So the quality of the data has increased astronomically, and we've really come a long way. We've actually updated. It's now released in Dorado. We have what's known as the Transformer Encoder Bot, which is the same type of platform that's run for ChatGPT. It's artificial intelligence that's actually used to help perform these base callings and help to increase our accuracy. And you can see here, just with the timeline, how things have changed gradually. And this is just in a matter of less than 2 years that we've gone from Q20 all the way to Q26. We also now offer through Dorado, and this really isn't for variant calling. This is more for de novo assembly. We have pre-assembly read correction with Dorado that can achieve up to Q40. So what we're having here is we're able to actually perform de novo assembly and increase the quality and confidence in the reads that are coming off of the instrument. We've also significantly improved our short variant calling. So historically, we've had lower indels coming through. We've increased our SNP accuracy, maintains at 99.9%, Q26 approaching that Q30. And we've made improvements to Claire 3 training process that will give us big improvements within our structural variant calling, which will help to improve our indel calling as well. Bacteria is one of our sweet spots. We're consistently hitting greater than Q50 for bacterial assemblies. And this just gives you an idea of the bacteria, how they have diverse modifications, how there's a range of native and synthetic data types that we actually use to train these base callers and improve our quality. And bacteria is really where we kind of hit our sweet spot in regards to quality. For plasmids, we offer a complete plasmid workflow. So our new Medaka model provides complementary improvements in a combination of recent base caller versions. So basically, we've updated our base callers. We're constantly updating Dorado. We're constantly updating our Minnow software. And that's one of the things that I know historically when I started at Illumina, the GA was constantly getting updates, constantly getting software updates, constantly getting chemistry updates. And it's just, we're doing the same thing now to improve that quality, to improve that yield. Our goal is to give you the highest quality we possibly can with the highest yield that we possibly can in the shortest amount of time. So if we look at the full suite, when we talk about quality scoring, we're looking at over Q20, and that's for high accuracy calling. So we have what's known as super accuracy, which will give you that Q26, and then high accuracy, which is usually good for pretty much everything that anyone is doing scientific wise. We are well above Q20 for hack. For soup, we're at about Q26. Pre-assembly for simplex read correction is at a Q40. And we've also been able to increase our structural variant calling as well, which has helped with our indel call. We've also made improvements to adaptive sampling. So now with adaptive sampling, you can run all 24 positions without any lag time, any type of regression. We've increased the sampling power by about 20%. And coming soon, you'll be able to base call on target only data and enabling the base caller quilt sequencing and sample and till. So you'll actually be able to put into the software how much data you want, and it will just run until you achieve that amount of data. So say, for instance, you're looking at a specific set of targets and you want 20 gigs. As soon as you reach that 20 gig for that specific set of samples, it will actually put the flow cell in a safe state, so you can save it in the refrigerator if you like to and use it at a later date, but you already have all the data that you need. So you're not continuing to run these samples if you don't need more than, however much data that you're looking for in the beginning. We're also making updates to Epitome, which we do frequently to add different pipelines, to add different workflows. The legacy cloud is going to be discontinued, and it will be read-only from now until the end of the year. And then we're going to have full integrated cloud functionality. So with Epitome desktop application, you'll be able to run it locally or on the cloud and pretty much have all of the same features that you had before. But this time you just have the option of doing it from one source, from the desktop application. This is now fully released. As I mentioned, you'll still be able to access the cloud for any of the older work, but it's only read-only now. And once you have the application, you have the choice of running locally or you can run in the cloud. So it just gives you ease of use, and it makes it easier for you to perform analysis. Devices and flow cells. So we have our P2 integrated, which is currently in early access testing. We have over 1,300 users that are currently using the P2 Solo. So we're just adding them to the P2 integrated family. Again, the P2 Solo doesn't have the computation with it. You can attach to the GridION or you can get a computer for it. We are, as I mentioned before, updating the Mark 1B to the Mark 1D. The Mark 1D, to me, it just looks cooler too. I guess just sleeker. You have better temperature control. It's easier installation and future compatibility with the iPad. So that's where you're going to I've actually seen the case, and it just sits right on top. The iPad plugs in, and you just put your Mark 1D right on top and get the sequencing. So this will be moving to early access in Q4. So if you went on our website and you registered your interest, you would be able to participate in early access or work with some of our earlier products. With that being said, we are going to continue life science and research products. We're going to continue that pipeline of beta testing, registered for early access, open early access, and then released. This has been the workflow pretty much ever since I've started, but now there are more and more requests from biopharma and other types of markets where they want our software locked down. They want to have notification like 6 months, a year in advance when we're going to make changes. So we've been listening. And what we've done is we currently have the Q-Line GridION. So the Q-Line GridION, it supports 21 CFR Part 11 compliance. It's on the R9 chemistry just because of the simple fact that we are only going to put on Q-Line products that are fully released and that have been vetted for several months. With that being said, the 2025 roadmap, we're going to add the PromethION 24 to that. And that's going to be on our R10, our latest chemistry. And then the GridION is going to have an upgrade. We're going to upgrade that to the R10, and we're going to also upgrade allow it to run RNA, because currently Q-Line cannot run RNA for the older model of the GridION. So you have the ease of use of if you're doing research and you don't really care if software gets updated and you really enjoy these changes and these advances in technology, or you can lock everything down if that's the type of environment that you need an instrument for. We're also, as I mentioned earlier, working hard to increase yield and accuracy. And what we have here is a new improved protocol and a new improved flow cell that actually increases the yield. So what we're seeing is that we're able to actually perform fewer loads. It's currently with our beta testers, and you're getting a yield of where you would see with the current flow cell. If you're performing the current protocol, you get about 100 to 120 gigs off of a flow cell. That is now bumped up to about 140. And what we're working on in R&D, we will be able to actually increase that to about 140 to 160. So you're getting about 140 to 160 gigs of data off of each PromethION flow cell. End-to-end workflows, when we talk about synthetic biology, we have a current workflow for mRNA QC, AAV, amplicons, and plasmids. I have a lot of biopharma customers that are currently using these applications and taking advantage of the full end-to-end workflows. For small genomes, we have 16S, which is currently in development, SARS-CoV-2, flu, RSV. RSV is going to be coming soon, but SARS-CoV-2 and flu are released and available. Metagenomics is also in development, and direct from colony sequencing is also released as well. And then we have our no-miss protocol. So that's the nanopore-only microbial isolate sequencing solution, and that's currently available as well. So we have complete workflows for bacteria, yeast, and fungi, and it achieves highly contiguous and highly accurate assembly. So it's really one of our sweet spots in regards to sequencing. And our end-to-end for human. Currently, we have our rapid whole genome sequencing in beta testing right now, where you can get 30 to 40x coverage for a whole human genome in less than 24 hours. You can go from sample to sequencing in about 4 hours. And if anyone's heard of Ewan Ashley's work out in Stanford, how he set the Guinness World Record for the fastest sequencing from sample to answer. It's one of the reasons why I do what I do, because it's very inspiring to hear the work that was done. So apparently a kid came in to the ER, he was coughing, they couldn't figure out what was wrong. They thought he had COVID. He did not. They basically ran his DNA on PromethION. They used all 48 flow cells. They went from sample to answer in like 6 or 8 hours. And they were able to find a rare variant, and he did a heart transplant. So they got him on the list, he got a heart transplant, and now today he's doing fine. So it just gives you an idea of the capabilities of this technology and how just fast sequencing, getting the data as quickly as you can actually help people. We also offer human workflows for cfDNA. So with cfDNA, you also get that methylation information. So you're getting, because it's the native cfDNA, tumor normal, and also single cell. So as I mentioned earlier, we have a full partnership with 10x Genomics. And we're also going to be getting full support for the GEMX protocol and the Visium spatial mapping protocol as well. You get about 80 to 90 million reads, cell-assigned reads, off of one PromethION flow cell for a 10x library. So you just basically, if anyone's ever run 10x, instead of performing the fragmentation prior to putting it on the Illumina sequencer, you keep those isoforms intact, and you just put them on our PromethION flow cell, and you get full isoform information. So you're getting 80% of the data that you would be missing with short read, because you're going outside of that 3 to 5 prime end. You're getting all of the isoform information. Also, further end-to-end workflows for human, we've partnered with Twist for a PGX program that's currently in beta testing. Teleseq has been fully released. Reduced representation methylation sequencing has been released as well. And human variation has been released as well. So in regards to human end-to-end workflows for large cohorts. So if anyone's ever run a full 24 sample run, and you may have one flow cell that falls off, what we've done is we're currently developing a protocol that's going to help with that and ensure that you get 30x coverage across all of your flow cells. So we have steps to prepare, to sequence, and to analyze. And basically the workflow is like this. You sequence and pool 24 samples across 24 flow cells, and you sequence all at one time. And what you'll see here is if you run 24 samples across 24 flow cells, you may have one drop off. With the newer protocol, when you've multiplexed those 24 samples, any lag is going to be picked up. So you'll get 30 to 40x coverage for every single library that you place onto the flow cells. What that will do is just to help ensure that you reach your designated goal of at least 30x coverage. Here's just a schematic where you're taking the 96 samples, you have a LIMS interface. Again, this would be, you could set this up to be fully automated, perform the sequencing on our instrumentation, and then perform the analysis via our Epitome suite. So this can all be attached to your LIMS, and this can all actually happen with minimal hands-on time. So when we also talk about whole human genome sequencing, we're talking about being able to detect modified bases, which include methylation. So I kind of put it like this. If you're in genomics, you get the methylation information for free. If you're in epigenomics, you get the DNA information for free. It's just there. You don't do any other type of library prep. You set up a normal run like you would for whole genome sequencing, but you're going to pick up all of the methylation information as the sequencing run is happening. All you have to do is turn on modified bases in the Minnow software when you start the run, and it will pick up the information for you. This is compared to our synthetic truth set. So as you can see here, we're getting 99.59% in regards to precision. Recall is about 99.42, and F1 is at 99.5. So compared to bisulfite whole genome sequencing, you get a higher number of CPGs called. You get a high correlation with whole genome bisulfite sequencing, and a single read accuracy of about 99%. And again, you're getting all of the information. And this plugs directly into IGV. So you can look at the information as you historically would if you were running it on another platform. And this just gives you an idea of what's available now. So live or post run, again, you can do this while it's actually running, or you can do this via Dorado after the run. And you have 5-methyl-C, 5-hydroxymethyl-C in CPGs, 5-methyl-C and 5-hydroxymethyl-C in all contacts, and 6-MA in all contacts. And offline, we have 5-methyl-C, 5-hydroxymethyl-C, 6-MA, and 4-MC. So this is what's currently available. And as I mentioned earlier, we continue to work on additional modified bases, and we're adding them to the list of what you can detect. So here's are some of the bases that are some of the modifications that are currently in development. And if anyone has something they're interested in, feel free to email myself or reach out to me or reach out to Eric or anyone else from our team, and we can see if it's on the list for R&D. So now I'm going to focus more on whole genome sequencing. I know everyone in this room knows that the human genome was not completed, back in 2007 when everyone said it was, that it actually occurred a couple of years ago, or not even a couple of years ago. And a lot of that work was performed by us. So we actually do offer a full T2T solution. You could do a nanopore only T2T run and assembly. And we've actually been able to interrogate that remaining 8%. So those dark regions, the Y chromosome. My wife tells me the Y chromosome is difficult for a good reason. So I can't disagree. So again, we've actually helped we've been instrumental in helping bringing that work forward. And personalized medicine starts with a complete picture. So if you're doing whole genome sequencing work, you want to have a complete picture. So 8% of the genome is missed by SBS chemistry. And it's actually, CHOP believes that it's more, that 19% of the genome is missed. Because again, you can't get into those dark regions, expansive repeats, things of that nature, structural variants, complicated regions of the genome, you just can't really get into. And decode genetics shows that short reads miss about 40% of all structural variants. Because again, you're taking 200 base pair fragments and trying to put them where the software thinks they belong. And in regards to methylation, less than 3% of all CPG sites are covered by standard genotyping arrays. That includes your epic arrays and things like that. So with our technology, you're able to open the door and get the answers of the complexities behind the biology that you're looking into. This is the work that I had mentioned earlier. So here's Ewan Ashley in his lab with the P48. And from DNA to report, it took less than 8 hours. Comparable SNP accuracy to short read sequencing. And this was right when KIT 14 was still in early access. This actually happened, I think, right around the time I started. And he was able to, as I mentioned earlier, go from sample to answer quickly and get results. And now they're working on cold cases. They're working with us to turn this into a protocol. Again, to run 48 flow cells would be extremely cost prohibitive. So we've dialed it back to about 2 to 3 flow cells. And that protocol is currently in beta testing right now. So you'll be able to quickly get a genome in less than 24 hours. Here's a study that was performed by CAR. This is an excellent paper. Happy to send it to anyone that wants to read it. Basically, what they were able to determine is that using a single PromethION flow cell, they can detect SNPs with F1 scores better than short read sequencing. So you can see here the data comparison in regards to the number of true variants. You can see the percentages right in line with Illumina. So, which is presumably the gold standard in sequencing accuracy. Bioinformatics, our goal is not just to increase yield and increase accuracy, it's to increase ease of use. I can personally say this is the easiest library prep I've ever done, and I've been in this industry for, as I mentioned earlier, for almost drinking age. The prep, we offer the reagents for that. You can fully automate the prep. So we work with Hamilton, we work with TCAN, we work with other vendors. The paper I mentioned earlier that was done by Card, they have fully automated their entire process. So they do all their sample prep on their Hamiltons. And we have a good relationship with pretty much all the vendors to help to build these protocols. If there's something on our website that you don't see we have a protocol for, just let us know. And we're happy to help out. We have a full automation team that can help you. Nanopore sequencing, obviously we provide the devices and the flow cells. And then for data analysis, we offer everything through Epitome. So if you were doing the whole human genome workflow, you would use the human variation workflow within Epitome. In regards to analysis, going back to Epitome, we have options for everyone. I know when I started, everything was command lined. I had to learn Linux. I had to learn all the command line stuff. You know, it's great that I know it, but I don't know if I'll ever use it again. But basically, we have an offer for everyone. So if you're just a scientist, you want to point and click, you want a GUI, you can take that route. If you want to actually perform more bioinformatic command lines, things of that nature, you can actually take that route as well. You can also introduce pipelines into Epitome. You can introduce your own workflows. You can modify workflows. So really, it's anything from the novice user all the way up to the experienced bioinformatician, which I know David is. Here's more information. There are QR codes here located if you wanted to get more information about Epitome. And you can also go to our website. This gives you an idea of the workflows that are currently available. So we have everything from live base calling, which is performed in our Minnow software, adaptive sampling, which is done in our Minnow software, and everything else in Epitome. So we have everything in Epitome from base calling all the way up to transcriptomics. So we offer pretty much everything from plasmid sequencing, metagenomics, whole genome sequencing, RNA sequencing, transcriptomics. Targeted sequencing. I'm going to geek out a bit because I'm kind of a nerd. The technology just amazes me. So with adaptive sampling, it's basically targeted sequencing without enrichment, without, PCR pull down, without, bead-based enrichment or anything like that. You put a bed file into the Minnow software. You tell it what target you're interested in. And what will happen is that as the DNA gets pulled down, as we saw in the video earlier, the DNA gets pulled down, it'll sequence 400 base pairs. If it doesn't match anything in your bed file, it will change the polarity and kick the strand out. So the motor enzyme's kicked off, so you don't have to worry about it being sequenced again. But it will then pick up another strand of DNA, and if it matches, it'll sequence it. So it does this, it reads 400 bases, which takes a second. So we'll read it and then kick it out, read it and kick it out. With our current updates in our algorithms for the PromethIONs, for the GridIONs, for Minnow, you can now do this on board. We've increased the speed at which you can do this. And for the integrated PromethION 2, you can actually do this on the instrument as well. This is a study that was done by Danny Miller. And basically what he did was to resolve any type of disease causing variants accurately via targeted sequencing. And if you read the full paper, you can actually see some of the clinical events where I think it jumped by almost 30 clinical events that he could not detect in short read sequencing. So just by adopting a targeted approach via long read sequencing, he was actually able to increase the amount of clinical events he was able to detect. You can also perform reduced representation methylation sequencing. So again, you're not doing any bisulfite conversion. So I know people in here have probably done some type of reduced representation bisulfite sequencing or some type of bisulfite setup with Zymo kits. It's a pain. It's a lot of work. You're damaging the DNA. You don't know exactly if things work properly. But with us, there's no change in the protocol. There's no damage to the DNA. There's no bisulfite conversion. And we've actually seen Chris Mason's lab up at Cornell in New York, where he's just doing like 5x sequencing for really good methylation data. Increasing our logic or adaptive sampling logic. This is kind of a slide to update what I was talking about earlier, where we've actually worked on the software. We've improved things within the software to optimize running all 24 positions for adaptive sampling. And you can see here, this is London Calling in 2023, just the amount of coverage you were able to get from a PromethION flow cell, how it's increased with adaptive sampling. This is going to be upcoming, where you're getting up to 337x coverage compared to the 200x coverage you were getting just a year ago. That's all I have. I'll be around for however long I need to be. And if anyone has any questions, please feel free to come up and ask, or if you have any questions now, I'm happy to answer them. Thank you. Thank you.
Edward, I'd like to see a de novo assembly as a. It's gonna be too. My own personal bias. Let me ask already. That one. I know you can do it for bacteria.
Bounty.
So I believe you just hit the arrow at the bottom. I don't think the down is working.
Running Nanopore is easier than running a laptop.
Especially when you're used to the app.
I know.
I know.
So what I'm going to talk about is a project we did with Kathy Gabrielson in comparative medicine. And she was interested in characterizing a rat osteosarcoma cell line. And we decided to give it a try using Nanopore. There we go.
I think so. Are we sharing?
Introduce yourself.
Oh, okay.
Are
For
we,
those online especially.
are we sharing? Can you see?
Yes, we're sharing.
Okay.
I mean, it's not, this is fine.
The host has stopped. I guess we're okay. Can you see online?
I can see.
Okay, we can't turn on the camera.
Do I turn? No, that's okay. Nobody needs to see me.
Alan's fine with that.
Okay.
Yeah, I'm Alan Scott. I'm a former director of the GRCF. So as I was saying, we wanted to characterize the cell line, and I thought the best way to do it would be using nanopore sequencing. So, so the problem with cell lines is that the longer you grow a cell line in culture, the more likely you are to get changes, particularly if you're working with cancer cells. So generally, the best practice for cell work is to have aliquots of the cell line which you thaw periodically. So rather than keeping a cell line going indefinitely, where it has the opportunity to accumulate more mutations, you characterize the early passage cells, and then you can keep going back to them, particularly if the phenotype changes. And about 10 or 15 years ago, there was an issue that people realized that a lot of cell lines that were being used in laboratories were actually not what they thought they were. In fact, a lot of them turned out to be HeLa cells. So the journals started requesting that laboratories characterize their cell lines so they knew exactly what they were working with. So we, the GRCF, started a service where we would do that. But you could only learn so much by looking at short tandem repeats, which is the way the methodology was done. And in this case, since Dr. Gabrielson wanted to characterize the cell line for other purposes, I thought it would be better to try to compare a couple of long DNA methods. And what we did was to use Nanopore to look at genome structure. We also used optical mapping from Bionanogenomics that's offered through the pathology group and Ying Zhao's group. And then we looked at expression. So we did short read Illumina sequencing for RNA from the cell line. And we compared it to public data that was available for osteoblasts, which are a related cell type. So here again is the summary. We used ultra-long reads on the cell line. So Nanopore offers something called that's a transposase kit, and you can get very long reads. The hardest part with using it, I think, is getting a high-quality DNA. So we had to futz around a little bit to get high-quality DNA from the cell line, but once we did, we were able to use the ULK kit to get very long reads. And then we compared that. Michelle back there did the prep for the standard prep, which was from blood from a Sprague-Dawley rat, the white lab rat. So the cell line was derived from a Sprague-Dawley animal back in the 70s. And we got the cells from ATCC, had them grown up, so we had fresh preps. On those fresh preps, Ying Zhao's lab did optical mapping, and we did the nanopore sequencing and the RNA-seq. And then when we got to the analysis part, we looked for, in the COSMIC database, which is something out of the Sanger Center, to look for what genes in humans are most likely to be characterized in osteosarcoma. And then we looked at those, the human osteosarcoma-related genes in the rats. And then we also looked at, for mutations, we looked at copy number differences, we looked at structural variants, we looked at methylation, and we looked at changes in RNA expression. And this is an example of what the optical mapping looked like. So what we found from optical mapping was that there was a large block of about 45 megabases on chromosome 7 centered around the MYC gene. The MYC gene is here at about 93 megabases. And you can see there's a lot of, there's a large block of amplification. When we did the same thing, when we compared that to the read depth from the Illumina, the Nanopore data, we saw something that paralleled the optical mapping very nicely. And when we looked at the agreement between the two methods in terms of copy number, this is the plot that we got. So there's a very nice correlation between what we saw from optical mapping depth and from nanopore sequence depth. And then here are this is the example of the top 20 cosmic genes for human osteosarcoma. We started manually going through, looking at these genes in the tumor versus the control animal. And this is an example of where we see the P53 mutation that's in all of the reads in the tumor, but absent in the control. And it turns out this P53 mutation is orthologous to a P53 mutation in humans that's known to be pathogenic. So it's pretty clear that this is pathogenic mutation. And the fact that it's fixed in all of the reads suggests, although it doesn't prove it, but it suggests that this was probably the initiating mutation for this tumor type. We then looked at some of the other genes that were listed in COSMIC. One of them was H3F3A, which is a known tumor suppressor. And what we found if you look at the highlighted reads here, we found a deletion. So this dark line represents the deletion. I left this line. And about a third of these reads show a deletion. So we suspect that there is less of this protein being made. Whereas you don't see that in the control. When we look at the region in more detail that I showed you before, you can see the copy number depth on chromosome 7. And if we zoom in on it, then you see these regions of what look like excess amplification. You see these little mesas or plateaus of higher copy number. And if we take a look at one of them in particular, so this here we're looking at a region of about 15 kb, and we can see some of the reads look like they're normal chromosome 7, like this one highlighted in black. So we're reading through the chromosome, whereas others look like they're truncated. So the nice thing about Nanopore is because the reads can be so long that you can identify secondary alignments from this data. And when we looked at the secondary alignments, some of these alignments from these regions were aligning to another location on the chromosome about 300 kb away. And some of the alignments are quite long. So this top one, for example, is 110 kb. So we have 110 kb of flanking sequence that aligns elsewhere on the chromosome. Here we have one that's about 55 kb. So that's the advantage of the ultra-long reads. You're not going to get that from Illumina. If you were using short read technology, you wouldn't be able to identify what's going on beyond that boundary, or it'd be very difficult to do it. So I think what's happening here is there's a translocation event between those two blocks of DNA, and we're losing roughly 300 kb in the middle. So what does this mean? What is going on in the cell line? We didn't do karyotyping, but this is a sample of what I think is probably happening. We're getting lots of small, probably double minute chromosomes or fragmented chromosomes. So in this case of these leukemia cells, you see all of these very small chromosomes. And I think that's what we're seeing in the osteosarcoma. And probably each of them has a copy of the MYC gene in it, which is giving a growth advantage to the cells that have excess MYC. Okay, we also looked at methylation. These are two more tumor suppressor genes. And here we're comparing the cell line to control. And you can see there's a difference in methylation patterns. The LSAMP is a large gene, it's about a 2 megabase gene. So it's spread out over several panels here. And CDKN2A is another commonly differentially methylated gene associated with cancers. When we looked at expression using RNA-seq and comparing it to the osteoblast data, we found that many of these genes in this cell line are overexpressed relative to osteoblasts. So everything that's highlighted in yellow is a gene where we had statistically significant more reads in the cell line than in the osteoblast controls. And you can also see from this list that many of the genes in this region on chromosome 7 have been identified in the literature as being associated with osteosarcoma. So that's what that's what OS marker means. So what that may mean is in human RCR sarcoma, the reason that these are markers is that it may not, it may be that in human disease, some of these same regions are also duplicated or have a higher copy number than expected, which is why these are considered marker genes. The other possibility is too that some of these other genes that are in that block of DNA centered around MYC may also contribute to osteosarcoma in some other way. And conclusions are the p53 mutation was probably the initial event, and then there were other events that were enhanced the tumorigenicity of the cell line. The amplified regions are likely heterogeneous blocks of perhaps double minute chromosomes. There are changes in methylation that may have already also affected tumorigenicity, and whether many of the osteosarcoma genes that are co-amplified with MYC is coincidental or implies some additional contribution, functional contribution to the tumor is something that remains to be answered, but is certainly interesting. So in terms of conclusions about long-read methods, long DNA methods are really valuable tools. We wouldn't be able to have sorted out what I showed you if we didn't have long reads. Optical mapping is great. It can identify large structural events. The minimum size that optical mapping uses are molecules of 150 kb. So anything less than 150 kb is ignored by the software. But still, nanopore sequencing, particularly the ultra-long sequencing, can identify changes that are large, but also you can find things at the single nucleotide level, like the p53 mutation that I showed you. And you can identify all these structural variants. And it provides for free the methylation data without additional manipulation, as Edward said. So when coupled with short-read RNA-seq, you can get a very thorough understanding of what is going on in a particular cell line. And I hope this represents a model for people who are working with cells, that for a few $1,000s, you can characterize your cells and have a very good understanding of what's going on. So if their phenotype changes, you can go back, do resequencing, and find out what happened. So anyway, this is the paper. It just came out in Genes, and I'll stop there. Okay.
We're going to take questions now.
Yeah, Edward, do you want to? We can both take questions.
Oh,
Are there any questions?
there are none posted to the Q&A at this time. So if you are an attendee, you're
Jim?
more than welcome to post to the Q&A or raise your hand, and I'll allow you to speak.
So about what depth of coverage did you achieve?
We got about 25x for the ultra long, and we got about 30 for the control animal. And the N50, which is sort of a measure of the median length of the molecules, for the tumor was about 78 kb, whereas for the control using standard methods, it was about 25, I believe, 25 kb. The reason we didn't do ultra long on the control animal is that there was a reference for rat, and we didn't think that it was necessary to do the additional work to get extremely long reads, because we weren't expecting a lot of translocation or copy number events in the normal animal as opposed to the tumor.
And that length is usually what you'll see, anywhere from 20 to 30 kb for just a standard one.
And the structural changes that you did identify, you have seen them with the shorter version, or you only would have seen them?
I think you probably would see them. They might be more difficult to interpret. So really what you're looking for are regions of overlap with another block of sequence. So that's why short reads don't work. If your read is 100 or 300 base pairs, say, the probability that you're going to be able to identify where that additional sequence maps is low, particularly if it's in a repeated region. So I think you probably would find it with the standard reads. It may be a little bit more difficult to find depending on the nature of the DNA at the ends of those positions. Does that make sense?
Yeah.
Yeah. Okay.
I think that wraps it up for the questions. We're going to close out, and I thank both Edward and Alan today for their presentations. Our next presentation will be October 23rd here in this same room, 11 a.m., and that will be featuring Illumina. And information and registration will go up sometime in the next week. So thank you all.